Okay, sir. You start the presentation, sir.
Yes, no problem. Yes, yes. Uh, so student is live.
Yes, sir.
Can you,
Student is
can you?
live.
Okay,
Attend the student.
okay, no problem. So myself, Dr. Anil Patel. Today, I am delivering you the special topic for the PhD coursework is a rational drug design. This particular topic based on the bioisostatic approach in the lead modifications. So, firstly, that what are the flow of drug design? So, drug development step is that first we have to identify the disease. Then we have to validate the target, how the disease can be arises. Then after we have to identify the particularly lead compound which can be validate this target and work as a that particularly lead molecules. After that, this compound have some property. We have to make that compound to the optimize as a lead optimization step. Up to the we have to remove their toxicity, improve the biological activity, as well as improve their solubility. We as well as improve their large step preparations. Then after it goes into the preclinical trials. Preclinical trials means in vitro and in vivo studies. So after that, in vitro and in vivo studies, compound goes into the phase clinical trials. We know that all three phase clinical trials, phase one, phase two, and phase three, in a human models. So these clinical trials result in the approved, the most potent drug to the market, approval and circulations. So these are the particularly drug development stage, so that in which we require lead molecules as well as we require lead optimizations. These two steps is very crucial to particularly clear the drug development step. So firstly that we are going to see the how the lead molecules can be identified. Then after we are going to see the lead molecule optimize. Especially we are taking the bioisostatic approach and drug designing approach. So that lead molecules. So lead compound is a compound that has a desired biological activity with therapeutic relevance. Means the compound gives the particular biological activity and a particular therapeutical response. Similar means antimicrobial activity, anti-cancer activity. So particularly lead compound in a drug discovery is an initial chemical hit which showing the promising biological activity against the target. Means it is our leading molecules to go discovery to the drug. So that lead compound is a initial chemical hits. So how we identify these lead compounds? There are certain process to identify these compounds. Firstly, that lead identification can be possible through the natural sources. Similarly, that natural compound can be work as a particularly in a pharmacological actions and identify their compounds, phytoconstituent present in the molecules, then go for the discovery. Then after it is a very currently useful the lead discovery through the random screening. Random screening means without regarding their structures, any compound goes into the check into the biological activity. That is called the lead discovery through the random screening. And this approach currently using as a particularly in a different institutes. And similarly, this approach used in NCI, National Cancer Institute, USA. That they are checking all the compounds without regard with their structure for the anti-cancer screenings. So particularly that HTS, high throughput screening approach, is a random screening approach without regard with their structure, all compound tested in the particular biological activity. Similarly, that third is lead discovery through non-random means it is a focused or targeted setting, means particularly compound designed for the particular biological activity. Maybe it's a homologous analog, maybe it's particularly in its analogs, or particular targeted focused targeted screenings. So this is a lead we can identify through the non-random approach, particularly compound designed by the particular for the particular biological activity. Then fourth one is a lead discovery through the drug metabolism study. Drug during the metabolism study means during the clinical observation in which the pharmacokinetic study. Pharmacokinetic means absorption, distribution, metabolism, and excretion study. So during this pharmacokinetic study, during the metabolism state, we can identify that the compound which is actually for the biological activity, because sometimes our drug goes into the body and it convert into the another molecules, means during the biosynthesis it can be converted into the another molecules, so that we can identify that which compound is active. And then after that we can identify the lead molecules. Lead molecules means the chemical hit which gives a promising biological activity. Then after, the fifth one is a lead discovery through the clinical observations. Clinical observations means that during the clinical trials, we can identify that compound's biological activity is due to the which particularly part of the molecules. That can be becomes our lead molecules, and that can be helped for the further drug discoveries. And then after, the lead discovery through the rational approaches means we can identify any compound by the rational approaches. Rational means focused and targeted approaches. Means we design a particular compound for the particular biological activity with particular parameters, structural parameters. So that these are the particular steps for the lead identifications or lead discovery. But we are going to see the further step, that how the after the lead identification, how we can modify the lead and how it helpful for the further drug discovery. So lead modification strategy, identification of firstly the active part of the molecules, means the pharmacophore. It means that lead compounds or any drug molecules contain a pharmacophore and oxophore. Which part we can call as a pharmacophore and which part we can call as a oxophore. So that particularly the part of drug molecules which is responsible for the biological activity, means the part of drug molecules which interact with receptor and which produce a pharmacological action known as a pharmacophore. Another part of this molecules known as a oxophore. So that first we identify the any molecules in a particularly containing a pharmacophore, then we can identify that which part is a pharmacophore and which part is a oxophoric part, so that we can modify that particularly oxophoric part and we can improve the biological responses. So this is the first one, that can be identification of pharmacophoric part and oxophoric part. Second one is a functional group modification. If we find a lead molecules and we can change functional groups, particularly modification, then it may be reduce their biological activity or it may be increase their potency. So that functional group modification means a functional group we can change. If fluorine group is present, we can change with another halogen, or hydrogen we can replace with the another functional groups. So it may be the lead modification strategy lead to the potent biological activity. After that, we can go through the SAR as well as QSAR, structural activity relationship and quantitative structural activity relationships. SAR means compounds structure and its relation with the biological responses. So this SAR, we can go for the SAR and we can check that which compound will be lead compound and it can be modified by the structural relationships. This can be done through the computational study also. This can be done through the computational study that is called the QSR. Okay, so this is a QSR part particularly. So this third one is a QSR strategy. Fourth one is we can prepare the privilege structures. Means the drug is already have certain parts of the structures, and we will make that structure similar to that drug. That is called the privilege structures, and this is called the drug-like molecules also. So we can prepare our compounds is similar to that particularly drugs already have in markets. So it is called a privilege structure as well as drug-like molecules. After that, the fifth one is a structural modification to increase the potency and the therapeutical index. So what here comes the potency, which means the potent, particularly molecules. So potency is a particularly strength of the biological activity. Means any molecules give the particular biological response, but however it is potent that we can check with their strength. That compound is a potent, or it can be a give us just biological responses, but it cannot be potent possible. So how we can increase their potency and their therapeutical index? Therapeutical index means the ratio of the drug concentration at which drug become a toxic and ratio of drug drug become a effective. So it is become particularly TD50 and ED50. It is ratio between TD50 and ED50 is called a therapeutical index. So particularly this can be gives their particularly how drug is safe. We can identify by the therapeutical index. Then after homologation approach can be useful for the lead modifications. Homologation means if we have a certain particularly part in the molecules is a CH2 groups, then we can make their higher homologous similar to the methanol, ethanol, butanol, that can be called as a higher homologous. So this homologous approach can be useful for the lead modification strategy. Homologous approach, certain types of improve in the particularly lipophilicity. This lipophilicity is an important part of the molecules for their absorptions. So that can be homologous approach can be useful to improve the pharmacokinetic property as well as pharmacodynamic property. Then after this homologous is a particularly means to the linear alkenes. If this homologous, this particularly is called linear alkenes. Now that chain branchings means we have prepared a propyl as well as isopropyl groups. So it is called a chain branching. It also improve their it also alter their pharmacokinetic and pharmacodynamic property. So this homologous approach can be useful as well as chain branching approach can be useful for the lead modifications, and which means it can be give the particular responses. Now another that's ring chain transformations. This approach can be useful to if we have a branched alkanes, then it can be converted into the particularly cyclic chain transformations. So these branched alkenes can be converted into particularly cyclic transformation called the ring chain transformation, and this strategy may be useful for the lead modification. Sometimes it gives a similar types of similar potency, and sometimes it can be alter the potency. So this can be helpful to resolve their pharmacokinetic as well as pharmacodynamic approach. But here I have highlighted that bioisosterisms. So we are going to particularly focus on the bioisosterism and other the drug discovery processes. Okay, so I am moving ahead for that particularly lead modification strategy in the particularly bioisosterism. So first, what we call the bioisosters. So bioisosterism is a procedure of the synthesis of natural analogs of a lead compounds by substitution of an atoms or in group or in the parent's compound for another with similar electronic and steric characteristic. So bioisosterisms can be useful to modify the lead molecules. So particularly Friedman defined that bioisosterism or its phenomena by which compound usually fit the broadest definition of isosters and possesses the same type of biological activity. So particularly bioisosterism is important in medicinal chemistry because it maintains the similar biological property. Means we are targeting if any cancer activity, then it's maintained their anti-cancer activity. It may be altered their potency, but it maintains similar types of biological activity. Similarly, it resolved the biological problems, means their potency, side effect, particularly their mode of actions, their duration of actions, and their biological responses. It can be resolved. So these types of work, several works as published by me, also my groups, as well as another groups. So bioisosterism is a lead modification approach that have been seen the useful to particularly alter their toxicity, modify the activity, or it can be alter the metabolism problems. It can be alter the metabolism problems. So this approach is very useful for the lead modifications. If we have a certain lead molecules, or we have identified certain lead molecules from the literature, then this approach can be helpful to design drug designs, new drug designs, which can be lead to the change in the potency of the molecules. So bioisosters means there are two types of bioisoster. First one is a classical bioisoster. Classical isoster means there are particularly outer most outer cells electrons remain the same. Means we can change the monovalent OH to monovalent NH2. We can change CH3 or any OR groups, ether or ester groups. Similarly, we can change the monovalent halogen with the different halogens. Divalent group means CH2 group alter with the oxygen, sulfur, selenium, means the valency remains the same. It is called a classical isosters. Similarly, we can prepare the trivalent alkyne, nitrine, nitrile, as well as we can prepare the tetravalent substitutions. So this is called the classical isoster. We can change in the molecule with the particular types of their valency with monovalent change with the monovalent atoms. Then it is called the classical bioisosters. Non-classical isosters means there are several types of examples. When ketone group is present, we can modify it to their acid, sulfonamide, acid amide, esters, or any amides. That is called the non-classical. Similarly, if we compound containing particularly halide groups, and then we can replace this halide with CF3, CN, these types of groups, then it is called the non-classical approach. Similarly, if compound containing a benzene ring and we replace this compound to the another heterocyclic ring, that is called a non-classical approach. So benzene ring replaced with another pyridine rings as well as another heteroatom containing ring, that is called a non-classical approach. So there are the several groups. Non-classical approach, that means we can their biological activity remains same, just changing in their structures. So these are the functional group we can change during the non-classical bioisoster. So here I have written some example of bioisosterism in a TNIP series, which is particularly in the market, currently in the market. Here that illotinibs hydrochlorides particularly useful for the non-small cancer, lung cancer cell line and pancreatic cell lines. So here we can say that it can be a particularly heteroatom containing compound, which is similar in the gafitinib. So this can be called their particularly heterocycle remains the same. Particularly pharmacophore remains the same in the molecule. Similarly, that can be a imatinib, that can be also that pyrimidine structures here, benzofuge pyrimidine structures. Similarly, that 4-atenib containing a heterocycle is a particularly red highlighted heterocycle is a quinoline rings. Here the sorafenib polar, particularly pyridine rings. And this urea linkers is produced. Similarly, the other urea linker can be prepared. The dasatinib. These all are the particularly examples of the changing of bioisosterisms, which are currently in the markets. These are just the examples. I will going to see the example of particularly modifications of the molecule as well as their strategy, how it is useful for the research work. There are similar examples in this slide also that containing a particularly heteroatoms which replace with another heteroatoms and which gives a particular anti-cancer activity, particularly anti-cancer activity. So these two pages containing the anti-cancer drug which is currently in the market is known as a TINIP series. So I have written here that a non-classical approach ring bioisosterism between the two different classes. So first one is an antibacterial class, which is a sulfadiazine drug, in which here the pyrimidine ring is present. That can be replaced with the isoxazoles. So this can be becomes a new drug, sulfametrozole, and which is a similar antibacterial activity. So these modifications is called the bioisosterisms. Here we have just changed this heterocyclic ring, that pyrimidine, to replace the isoxazole. So that can be oxazole and isoxazole derivatives that can be give the that bioisosterism approach. Similarly, we can see the second example in the celecoxib series. Here we can see that anti-inflammatory drug celecoxib, which containing a particularly pyrazole is a heterocyclic core. If we replace this pyrazole into the isoxazole, and that can be converted into the validacoxib, which is similar anti-inflammatory activity. In the third molecules that have changed to particularly by isosterism that replace this benzene ring to the pyridine ring, as well as this isoxazole ring to the pyridine rings, and that can be also itoricoxibs. So these all three molecules are designed through the bioisosterisms. Then we can see the another example is an analgesic drug. That nicotine have the very good analgesic activity. We know that. So this pyridine replaced by the particularly furan ring systems, and that is give the also analgesic property. This is published in the current medicinal chemistry in journals. So this approach is very useful to design similar molecules with similar biological activity. So this is called the bioisosterism. Now another example is the particularly ranitidine. These types of structures, it can be ranitidines. Here just change that furan replaced with the imidazole, so it becomes a nizatidine, and these molecules also a similar anti-ulcerative activity. We know that ulcerative activity, that ulcer means that can be create infections, stomach infections. At the time we are taking the anti-ulcer drug. So when the doctor prescribe antibiotic, at that time it also prescribed anti-ulcerative drugs, ranitidine, omeprazole, that types of molecules. So these transformations is called the non-classical bioisosterism approach. Here the furan ring system replaced by the imidazole ring systems. So these types of drug design strategy helpful for the further lead discovery or further drug discovery. Similarly, we have three class of antihistamine. Here we can say that this is known cyclics, particularly dimethylamine, and this is also known cyclic ethylamines. Then after this diethylamine, particularly converted into the ring chain transformation to the pyrrolidine rings. So this is called the ring chain transformations or chain to ring or ring to chain transformation, and this compound has twice activity of C and many times greater than the B. So this ring chain transformation lead the more potency. Compound A has a twice activity of C and it is a many times greater potency than the B. So this approach can be useful for the lead modification. If we found the list lead compounds, if we found if we found the lead compound have containing these types of group, then we can alter this type group to the these types of group. This is called the ring chain transformations, and this may be help to improve their biological activity. Here is an example of antihistamine activity. Next, in example of bioisoster is aminopyrin. Aminopyrin is a marketed analgesic drug and it approved in 1896, and in 1920 it was relieved from the market because it's a carcinogen because it produces toxicity. So the Roche group in 1951 they particularly modified this particularly NN-dimethyl group to the isopropyl group. NN-dimethyl group to the isopropyl groups. And these types of bioisosterism modification remove their carcinogenic actions. Means detoxification can be done through this changing of the particularly groups. So their toxicity removed and it can be available in the market as phenazone. So how the bioisosterism approach can lead to the particularly improve their potency or as well as improve their reduce their toxicity. So these are the particularly example of a marketed drug. Now we are particularly going to focus on the particularly anti-cancer agent because the next example I have it is particularly on anti-cancer agents. So currently I am going to discuss about the classification of anti-cancer agent or cytotoxic agents. Particularly broadly classified the classification as a four class that alkylating agents, antimetabolites, mitotic inhibitors as well as topoisomerase inhibitors. So cytotoxic agent or anti-cancer agent, that molecule that permanently attach to the DNA. It means the molecules permanently attached to the DNA with restoring its shape, and particularly it damage the DNA and improve work as a cytotoxic agent. Means example we have that cisplatin that can be worked through the DNA alkylating agent that can be alkylate DNA. It may be through the intrastrand cross-linking or single strand cross-linking. Similar to the cisplatin, there are certain types of nitrogen phosphate reported as a alkylating agents, and that can be also cross-linked with the DNA and produce the anti-cancer activity. Similarly, that B-hydroxymethyl derivative. So I have highlighted here that N-mustard as well as B-hydroxymethyl derivatives because I will going to study. I will going to see you that how the nitrogen mustard and B-hydroxypyrrole derivatives can be prepared as a new drug designs by using bioisosteric approach. So that's why I have highlighted here. The second is anti-metabolite, molecule that appear to the nucleotides and so are incorporated into DNA, which leading to non-functional DNA. So that's why these 5-fluorouracils, these types of molecule can be work as a anti-metabolites. Mitotic inhibitors, so molecules that block cell division by preventing microtubule functions. That is conotubulin polymerizations that can be worked as a microtubules are the vital for cell divisions. The main example are the vinca alkaloids, then vinblastine, then vincristines. These types of alkaloids can be worked through the mitotic inhibitors, and that can be produce anti-cancer activity. The fourth one is can be enzymes that can be topoisomerase inhibitors. Topoisomerase are essential enzymes that maintain the topology of DNA. That particularly maintains the topology of DNA. So that inhibition, that type 1 topoisomerase as well as type 2 topoisomerase, with both can be a transcription and replication of DNA by the upper setting of proper DNA supercoilings. For example, that irinotecan or topotecan that can be worked through the topoisomerase inhibitions. So mainly I am going to focus here the 4 class. There are the several class of the classification of anti-cancer agent. So I have written here the nitrogen mustard. So I am going to present here that one of the compound is a uridomustins, is a water soluble and potent anti-cancer agents. This is published in first molecule is published in 2011 in bioorganic medicinal chemistry by the Novel Kapuria and Sang Bamsu and its groups. Because I worked there in Academia Sinica, so these compounds become lead molecules. These compounds BO1055 becomes a lead molecules because there in the literature there is not only one publications in the for the BO1055. It has almost 2 patent as well as 7 publications in the very reputed journal in the oncology, several journals. So that uridomustine becomes a lead molecules for the further research. So in our group, Satish Kumar Tara has designed molecules by using their bioisosteroid approach. He particularly altered this amide, this particularly amide is a benzene alkyl amide conjugates. Here we can see that here CONH group is there, and in the second this part BO2094 here the NHCO group is there. So particularly just altering the position of carbonyl and nitrogen, amide and enilides, then it becomes a more similar potent, which is not for more potent but similar potent molecules. So these types of approach is called the bioisoster approach, and we can see that these types of nitrogen mustard can be the monoalkylation with the DNA, as well as it can be intercalations, as well as intrastate cross-linking, as well as interstate cross-linking is possible. So these types of studied particularly in this paper, and the Satish Tara has published this paper in the European Journal of Medicinal Chemistry in 2014. So these types of approach, bioisoster approach, can be helpful to design new molecules with the similar class of activity as well as a more potency. So this compound is a BO1055 also is a water soluble compound, as well as BO2094 is a water soluble. Here water soluble is the most important because we know that if molecules in our body, the first step is absorptions. If molecules does not have that particular solubility, that cannot be absorbed in our cell membranes. So they have particular partitions, water solubility as well as lipophilicity required to get a particularly concentration in the blood circulating systems. So these two molecules have very good solubility, water solubility, and that can be gives a very good biological activity. However, there is a disadvantage of this nitrogen mustard is a toxicity to the normal cell line. Normal cell line. This nitrogen mustard have a toxicity to the normal cell lines. So these compounds give the very good cytotoxicity against various cancer cell line. However, this compound have a disadvantage that it gives a toxicity to the normal cell line also, normal human cell lines. So we have replaced particularly this particularly types of work to the bis-hydroxypyrrole works. Now we are moving ahead to the structure of some other DNA alkylating agent similar to the particularly nitrogen mustard derivatives. So here our first is a mitomycin C, MCC, is already have a anti-cancer activity. So these are the particularly groups. I am focusing here, going to focus here compound number 4 and compound number 5. That compound number 4 and 5 gives a have the OH groups which can be interconnect with the DNA and produce a anti-cancer activity. So I have some mechanisms of mitomycin C. We can see how the mitomycin C can be work as a DNA alkylating agent. Then after we can design some molecules with the similar functionality. So we are going to see the mechanism of mitomycin C. Here we can see that mitomycin C, when as a result, it can be cross-linked with the DNA and produce a DNA cross-link edit. Here the mitomycin particularly give the DNA cross-linking edit. So reduce activation and DNA cross-linking by the MMC take place. So this MMC activity can be possible in another molecules with the pyrrole. We have identified that this pyrrole can be possible due to DNA cross-linking activity. So we have particularly proposed the mechanism of DNA cross-linking by these bis-hydroxypyrroles. Here we can take the diols as well as carbamates, or isopropyl carbamates or methyl carbamates or ethyl carbamates. So these types of compounds particularly give the mechanisms with the DNA and that can be interact with the DNA and that produce a DNA cross-linkings. How it can be confirmed? So this can be confirmed by isolating the IDECT and by confirming their structures. So these types of particularly reports, Anderson and groups has a report that particularly compounds. One compound is that particularly IPP. Here that IPP that can be when interact with the DNA, it can be produce a DNA cross-linking. Here we can say that this is a DNA adduct of the pyrrolizolin derivative, particularly compound 18. These compounds come from this IPP. When IPP treated with the DNA during the DNA cross-linking study, it can be bind with the DNA and it can be produce these types of anti-cancer activity. So these types of adduct can be possible. It is a part of DNA. It is a part of DNA. And this compound interact with the DNA and produce these types of alkylation, DNA alkylations. So compound 18 work as a DNA alkylation by particularly these types of groups present in the molecules. So this bis-hydroxypyrrole, particularly bis-hydroxypyrrole derivative or particularly bis-hydroxypyrrole ethyl carbamate or isopropyl carbonate or methyl carbamate can easily prepare these types of DNA adapt and produce anti-cancer activity through the DNA cross-linkings. So here the monoalkylation I have represented the particularly in interstrain DNA cross-linkings as a dialkylations. So these types of mechanism which is similar in a cisplatin as well as in a nitrogen mustard, which gives a similar types of actions in the bis-hydroxypyroles. So these compounds become these types of compounds become a lead molecule for us to design several molecules. I will show you there are certain types of molecules reported by our groups as well as other groups. So we can so here we can see that a scheme 1, that is particularly bis-hydroxypyrrolidine derivative and their other analogs. So these types of particularly groups is present in the mitomycin C. After that, this compound is imidazole containing this carbamate, methyl carbamate. Here that pyrrole containing particularly diarrhea carbamate, compound number 2, class number 2. Class number 3 is particularly IPP, we've already seen. Then after class number 4 is particularly furan thiophene ring systems fused with particularly pyrrole systems. Similarly, compound number 5, pyrrolidine is compound number 6. Similarly, that particularly heterocyclic bindings. But here I have represent to the compound number xi means tens and 11, 12. So we can I have highlighted here particularly two molecules because these the molecules prepared by me and my groups. So I am highlighting here that particularly this compound and these compounds. So this compound is basically designed from this bis-hydroxypyrrole derivatives. I will show you that how the compounds gives a particularly activity and particular responses. So bis-hydroxy first is a bis-hydroxymethylpyrrolothalazines. So in we see here that particularly it contains two types of pharmacophore, thalazine as well as bis-hydroxypyrrole. This bis-hydroxypyrroles we have taken as a lead molecules in these several references. These several references reported that due to the presence of these bis-hydroxypyrroles, it produces anti-cancer activity. So we have several references also that thalazine is very useful as a pharmacokinetic and pharmacodynamically pharmacophore for the anti-cancer activity. So we have combined these particularly groups thalazine as well as bis-hydroxypyrrole, and we prepare the hybrid molecules as a pyrrolothalazine bis-hydroxymethylpyrrolothalazines. So how these molecules can be synthesized? First, I have just represented design, how we have designed these types of molecules. Bis-hydroxymethylpyrrolothalazines. Now, how this is just the theoretical design. How the synthesis is possible to prepare these types of molecules. So I am going to here show you the scheme, how it's work. So firstly, we have taken as a chlorothalazine as a starting material. Then after it react with certain types of secondary amines in the presence of TA and ethanol, it converted into the amine substituted thalazines. Then after we have reacted compound 26 with particularly TMSCN in the presence of Lewis acid as a AlCl3 and acetyl chloride. So it can be give the particularly cyanogen at the nearest position of the nitrogen as well as acetylations. So this N-acetyl and their particularly position as the 2-cyano, which is helpful to give the disalder reactions and the cyclization reactions. So this reaction can be formation through the intermediate as a HBF for salt. This salt is prepared in the presence of acetic acid at 60 degree. Simultaneously, it can be converted into the cyclic pyrrole structure by using a dimethyl acetylene particular DMAD in the DMF 100 degree. So acetylene dicarboxylate can be converted into that particular diester as a 20A2D. Then this diester is reduced in the presence of lithium aluminum hydride in ether and dichloromethane solvent and produced to diodes. Now this is also our targeted molecules. But we can prepare that in another living groups as preparing their carbamates. So this compound 29A2D reacted with the particularly isocyanates. Maybe we have used here ethyl and isopropyl isocyanate because the methyl isocyanate gives a little bit more toxicity. So we have used here ethyl and isopropyl isocyanate in the presence of TA and DMF. It converted into the carbamates. Now the important part is their biological response. You can see that particularly you can see that compound 29D means that by piperidine structure. Bipiperidine structure at these positions R1 that gives a very good potency in the HCT116 means a colon cancer cell line, as well as S46 means a non-small cell lung cancer cell line. It also gives a very good potency in a particular H526 means a small cell lung carcinoma, as well as it gives in very good potency in PACA S1 means PACA S1 is a pancreatic cancer cell lines. So we have compared this compound with the standard drug as a cisplatin. So you can see that it is a far more potent, means almost 20 to 40 times more potent than the cisplatin in all four cancer cell lines. So it means these types of compound give a very good potency. These compounds, particularly 29D, is a more potent than the cisplatin, 20 to 40 times more potent than the cisplatin. So we go further that particularly how it's after that we have checked that we already seen that how can it can be interstand or intrastant with the DNA. So this is a mechanism which we have already seen earlier, that these types of particularly hydroxy group or carbamide group when interact with the nucleophile as a DNA, it can be produce a single strain DNA linking, as well as it produce a double strand DNA cross linkings. So that possible mechanism we have seen through the SN1 electrophilic reaction of particular B-hydroxymethyl pyrrole with the DNA. So these types of mechanism which is similar to that particularly cisplatin, which is similar to the mitomycin C. So that our most potent compound we have checked for the DNA interstrand cross linkings. DNA interstrand cross linkings. We have taken as a melphalan as a standard drug, and we have taken our compound in 0.1, 0.25, 0.5 and 1 micromolars. Here we have taken melphalan as a 1 and 5 micromolars. We can see that here the single strain linking, cross linking, as well as intercalating with the DNA. We observed this intercalating in the particularly our 29D compound is more with as compared with the melphalan drugs. So that it can be used that our compound is in DNA interstrand cross linkings. So this we have checked through the electrophoresis in a particularly and we have taken a control which can be due to single stain. Then after it can be, we can see that the melphalan is a 5 micromolar, see the single as well as DNA intercalating, which is, we found that this 1 micromolar of our compound shows a very strong DNA interstrand cross-linkings. We can see that 1 micromolar of the particularly 29D, so the very strong DNA interstrand cross-linkings. So that via this mechanism, we found that this compound is a very good in DNA interstrand cross-linking. As well, we have checked their in vitro, in vivo activity. So that this can be gives a very good biological activity. So we can see that this particularly work we have published in a Journal of Medicinal Chemistry, and you can see that this particularly thalazine hybrid with the bis-hydroxypyrrole produce a very good DNA interstrand cross-linking, and it can be inhibit angiogenesis and induce a DNA intrastrand cross-linking. So this work we have published in 2019 in the Journal of Medicinal Chemistry. After this work, after this work, we have designed with this compound we have taken as a lead molecules, and we have designed new molecules which particularly more focusing on particularly their lipophilic part. Means this compound have a very good activity, however the problem is their lipophilicity. Problem is their lipophilicity. So we have designed the particular molecules to improve their lipophilic property. So in the next class, to alter this lipophilicity, we have designed this class of compound, means this benzology approach we have used. You can see that we have just replaced here only one benzene is present. We have attached one benzene more and prepare that benzopyrrolothalazine. This is a benzopyrrolothalazine hybrids. So just we have replaced this two hydrogen to these particularly aromatic ring systems. So molecule designed by the benzology approach. Similar, we have taken that bioisoster approach. So these benzology molecules alter their lipophilicity without losing their potency. Uh, this paper we have already published in a JMC, as well as we have taken a patent. My guide Dr. Sang Long Su have already taken this molecule as a patent. Then after we have modified this benzology approach, we can say that how it's improve their biological activity and we can alter their lipophilicity. So how we can synthesize? Similar to that particularly previous synthesis, but we have taken here the particularly anhydride. This anhydride converted into thalazines. This thalazine converted into their dichlorothalazines, benzo dichlorothalazines. So this particularly compound 14. After that, we have replaced one halogen, means a partial halogen replacement with the amines. Partial halogen replaced with the amines. We have taken the amine as a particularly secondary amine, that dimethylamine, particularly pyrrolidine, piperidine, as well as morpholine, as well as particularly bipyridine. Because bipyridine we have taken as a reference from our earlier paper, that already that bipyridine compound gives a more potency. So we have also taken that bipyridine here as a reference. So we have prepared that one chloro and Then this chlorine is replaced in the 16A compound by treating with their palladium carbon and methanol and reflux conditions. So this becomes similar to the earlier class of compounds. And then after which is we have treated this compound 16 with HBF4 in acetic acid to give a HBF4 salt, which can be work as a starting material of diacyclic reactions. So that can be a dienes, and that dienophile is a dimethyl acetylene dicarboxylate DMAD that can be produced with particularly types of esters. This 18th compound ester, which is reduced to the particularly lithium aluminum hydride, which converted into the particularly B-hydroxypyrrole derivatives. Sorry. This B-hydroxypyrrole derivative, and this B-hydroxypyrrole when other treated with ethyl isocyanate in TEA, they produce a particularly ethyl carbamates. So this is a particularly scheme for the particularly designs. We have first designed these molecules, then after we have checked that how it can be synthesized. So this way it is possible to synthesize. So we have prepared that 19A to E as a diol and 20A to E as a ethyl carbamate. Now we have checked this compound in cytotoxicity. So we have checked these all compounds in the cytotoxicity against particularly colon cancer cell line, colorectal HCT116, as well as we have checked is a pancreatic cancer cell line, PACA-S1, as well as renal cancer cell line 7860. So we can see that we have here taken three standard cisplatin, irinotecan, as well as sorafenib. And we can see that 19A and 19B both gives a more potency in all three cancer cell lines. We have checked this compound in a 8 cell line, but however I have written here 3 more potency for the three cancer cell lines. We can see that these compounds 90A and 90B they give the similar concentration, but which gives a more potency is a 0.93 micromolar as well as 0.65 micromolar. Similar to the renal cancer cell line, it produces a 0.37 micromolar as well as 0.39 micromolar. So these compounds 90A and 90B is more potent compared with the cisplatin, irinotecan as well as sorafenib. We found this compound gives a very good biological response in a various cell lines. So we have checked their mechanisms similar to the earlier that how it can be intercalating with possible with the DNA. So benzopyrrolothalazine hybrids, we have checked alkaline gel electrophoretic analysis. This is electrophoretic analysis. And here we have taken 19A to E compound at 37 degree for 2 hours for the various concentration with the plasmid DNA. Then after the linearization and digestion by the particularly the DNA was denatured under alkaline condition. The intra-strand cross-linking and single-strand DNA molecule were electrophoretically separated on alkaline gel. So this is look like similar to the TLC in alkaline gel. So we have taken as a melphalan as a standard drug as a positive control. So we can see that 19A, 19B, 19C, 19D, all 4 compounds gives a very good interest and cross-linking with the DNA. We can say that this melphalan 5 micromolar gives these types of cross-linking, whereas our molecules 19A gives these types of interest and cross-linking. Similarly, in a 1 micromolar compound gives 19B, it's a very good interest and cross-linkings. So that interest and cross-linking studies through the alkaline gel for electrophoretic analysis gives a very good result that produce a DNA cross-linkings. So nextly, means these types of compounds we have published in 2021 again in a JMC Journal of Medicinal Chemistry. So this benzologic approach gives a very good lipophilicity as well as it gives a very good potency in angiogenesis through the anti-cancer activity. So it inhibits angiogenesis and induces DNA cross-linking. So they have this particularly compounds we have given to the mice model in vivo as a oral drug. We have provided that injectable as a oral drug, so that can be a particularly oral anti-cancer compounds. So this compound gives a very good activity. So this approach, this means this benzology approach gives a very good result. Now I am going to see here another these types of drug design we have done in a previous work, that can be that particularly pyrrolophenanthridine. So we have taken here the pharmacophore is a phenanthridine, and again the most active pharmacophore DNA interested in cross-linking is a bis-hydroxypyrrole. So we have combined this particularly to produce that bis-hydroxymethylpyrrolophenanthridine, these types of molecules. So we have designed this molecule as a class 1. Similarly, we have combined this bis-hydroxypyrrole 2, particularly dibenzoisoquinoline. This dibenzoisoquinoline molecules, because this dibenzoisoquinoline types of compound is already reported in the literature as a natural products, and which also gives a very good anti-cancer activity. So we have combining these two pharmacophore, and we have prepared that dibenzopyrroloisoquinoline is a class 2. So these designings come from that previous work, our previous work. We have already reported in JMC in 2019. We have already get the preliminary data, and then after we have started the designing of these types of molecules. So these types of work is similar to the previous work, but we get very good result. So initially the synthesis part. So we have taken a phenanthridine as a starting material. Then after we have treated with the bromoacetic acid to produce a bromoacetic acid salt, and which we treated with particularly DMAD, acetylene dicarboxylate. Then it produces ester, but without here substitutions. R1 is equal to H when we are treated with the 18A. Then after, if we are treated this compound with particularly our previous chemistry is a TMSCN, trimethylsilicyanide, in the presence of Lewis AlCl3, it produced that particularly N-acetylated alpha cyano product, 2 position as a cyane product. Then after we have earlier reported the chemistry of preparing the HBF4 salt, and which can be a precursor of Diels-Alder reactor. This react with the DMAD producer dicarboxylate. So in the first, particularly for the first route, we have prepared here hydrogen. In the second route, we have prepared here the substitution. B to F we have prepared to this particularly this route, and particularly A prepared from the first route by preparing the bromoacetic acid salt. So that particularly after that, this diester which reduce to particular diol by using a lithium aluminum hydride in ether and dichloromethanes. And then after it can be converted into that particularly ethyl and isopropyl carbamate, which we earlier prepared in the earlier papers. So chemistry is almost similar, but we are more focusing towards the biological activity, because we have prepared the similar analogs, similar bioisosters, similar approaches we have taken for the drug designings. So we are focusing on the biological activity. Now, this is of class 1 compound. Here we have reported class 1 and class 2. So, this particularly route for the class 1. Now, we have taken a different chemistry for the class 2. So, these class 2 compounds we have designed, but particularly required a very good chemistry. So, we have started with the particularly phenanthrene aldehyde, which can be converted into which can be reduced to their alcohol by using a sodium borohydrate. Then after this particularly alcohol converted into a halogen by PBr3, means it's bromo derivative PBr3 in dichloromethanes. This particularly bromo derivative react with the diphenyl glycine ethyl ester, so that this compound is very important to produce amino acids, amino acid derivatives. So that diphenyl methylene glycine ethyl ester, it react with this bromine in active methylene positions. Then it produce a compound number 27. After that, this compound number 27 is treated with particularly hydrochloric acid. So this particularly benzophenone, diphenyl ketone can be reduced, and this diphenyl ketone reduced to produce this NH2 groups as a hydrochloride salt. This now NH2 groups gives a particularly reaction, cyclization reaction in the presence of formaldehyde. Means it's types of Bissler-Naprosky or particularly isoquinoline synthesis reactions. So this NH2 group cyclize to particularly isoquinoline derivative. So dibenzoisoquinoline derivative is produced as a compound 29. This compound 29 further treated with acetyl derivative, acetyl chloride or acetyl anhydride, acetic anhydride in the presence of T and THA produced the N-acetyl derivatives. Now this N-acetyl derivative possibly directly convert to diester, but here we have converted this ester to the acid by the hydrolysis in the presence of ethanol and one normal NaOH. So ester is hydrolyzed to the acid. Now this acid and N-acetyl compound of 31A to E treated with the particularly acetylene dicarboxylate, dimethyl acetylene dicarboxylate to produce this bis hydroxy. So means bis diester. Sorry, it diester 32A to E. Now this diester further reduced to their diol. Here we have just prepared the 5 compound. Due to their high lipophilicity and solubility, we have restricted this compound up to the 5 molecules, because these compounds becomes very bad when we increase the aromatic part here, then it reduces their solubility. So that is very problematic to check their biological response. So we have restricted this compound up to the 5 molecules. Here we have prepared that particularly 12 molecules diol as well as carbamates. So here we have synthesized more molecules as 18, 6 for diol, 6 for ethyl carbamate, and 6 for isopropyl. Total 18 molecules we have prepared for this class 1, and 5 molecules we have prepared from the class 2. So we have checked all the compound for the biological responses. all the compound for the biological activity. So we can see that here we have taken that at the cancer cell line HCT116 means it is a colon cancer cell line. H1650 means it is a small cell lung cancer carcinoma. H460 is a non-small cell lung cancer cell line, and PACA S1 as a particularly pancreatic cancer cell lines. So I have written here just four compound that 21A and 21B as well as the 31A and 31B. We have compared this all four compound with the C splitting, and we found that this four compound gives a very good potency in all four cell lines comparison with the standard drug cisplatin's. So then after that we have checked that particularly uh, anti-cancer activity for that mechanism. We have checked this compound for the DNA cross-linking as usual, similar to other. We have checked that this compound. So we can see that compound 21A, 21B gives a little bit cross-linking with the 40 micromolar solutions. Compound 21 gives a very good, particularly inter-strand cross-linking to the particularly 20 micromolars, whereas the compound 33A and 33B very good inter-strand cross-linkings comparison with the melphalan. So particularly we found that the compound 31 and 31B is very good potency in a DNA cross-linking. However, the compound solubility is very bad for the in vivo screenings. So we have taken the compound 21A for the further anti-cancer screenings. So every time we have checked this mechanism through the cross-linking DNA cross-linkings. We have checked that single standard DNA. We have taken that DNA for the electrophoretic analysis, and we have checked that compound is interact with the DNA, how it can be cross-linked with the DNA. So this result give the idea about our molecules with interconnect with the DNA. So this is the first mechanism study. Then after we have checked these molecules for the cell cycle inhibitions. So we have take this study for the different concentration of the molecules, means without compound, and we have checked this compound 2.75 micromolar, 1.5 to 12 micromolar. Means in a different concentration we have checked for the 24 hours as well as 48 hours as well as 72 hours. So this cell cycle inhibition study gives a particularly idea about DNA interacting or DNA damaging. How it can be cell inhibition is possible through this particularly cell cycle inhibitions. So we can see that 21A compounds, particularly cell cycle progression predominantly at G2M phase. Therefore, compound 21A on cell cycle progression was further investigated in particularly H46 cells. Now 21A compound first goes in G2M phase. First goes in the G2M phase, and then after it can be after 24-hour arrest, it in a G1 arrest at 72. So we can say that in 72 hours, after the 72 hours, 77% particularly in a G1 phase, means that can be gives idea about the induced several DNA damage, and particularly that can be inhibit the cell cycles. So These types of cell cycle inhibition study give the idea about how the drug can be interact with the DNA, and it can be DNA interacting agents. So this is our further, this mechanism we have already studied in other two papers also, but I am showing here only because this is one examples which we have studied in all papers. So these types of cell cycle inhibition take place, and it gives our particularly response for the particular biological activity, and this gives an idea about the particularly cell DNA damagings. Now that this is a one types of mechanisms, we have checked DNA cross-linking, we have checked the cell cycle inhibitions. Then after we have also checked that apoptotic cells death. So apoptosis we have checked, but we found that our compound 21A, it gives a little bit cross linking, but it is a more power, more than the particularly melphalan, but we have taken as a higher concentration as 40 micromolar than the standard one. So this we have checked for the further study of the cell cycle inhibition, and we have further study for the apoptosis. So here that to confirm that compound one trigger the apoptotic cell death, we have checked that apoptosis analysis, and we have performed an annexin B binding assay. So here we have taken that at H46, particularly non-small cell lung cancer cell lines, with treated compound 20A of various concentration. Here we have taken a concentration as a 1.5 micromolar, 3 micromolar, and 6 micromolar. And we have taken a standard drug as a cisplatin as a 10 micromolar. So we can see that here the particularly cells then with this compound subjected to the flow cytometric analysis, as we seen in the particularly figure 1, that the compound have significant time dependent increase in percentage of particular V cell after 48 hours of treatments. So that it can be seen that 6 micromolar compound, which is similar to the that cisplatin of 10 micromolar compounds. See 10 micromolar concentrations. So that frequency of energy we shall summarize in the figure B, that we can see that 6 molar compound, 6 molar concentration of compound 21A is almost apoptotic cell concentration is more than cisplatins. So that can be confirmed that trigger the apoptotic cell death of the compound 20A by this apoptosis analysis. Apoptosis analysis. So these all three types of mechanism we have performed in a particularly to see the DNA intestine cross-linking as well as cancer cell deaths. After that, we have particularly checked that in vivo activity. As we know that in vivo means that there is a activity in the mice model study. So we have taken a compound 20A, 21A is a dose of 2 cycles. We have taken oxaliplatin is a standard drug. We have taken oxaliplatin dose as 7.5 milligram per kilogram of mice, and once per week for 2 cycles. We have taken in vivo, we have checked that compound, particularly H46 means a non-small cancer lung cancer cell line, as well as HCT116 that can be particularly colon cancer cell lines. So we have checked the in vivo study in these particularly types of cells, H460 as well as HCT116 xenograft models. So we have first induced cancer in a mice model, and then after we have checked that how cytotoxicity of the compound 21A. So here that particularly A is a average tumor size, and particularly figure B is a body weight change. How the body weight change? So here we can see that particularly compound tape 21A suppressed approximately 66 percentage as well as 72 percentage. 66 percentage in a particularly H46 cells mice model xenograft, as well as 72 percentage in HCT116 xenografts. So it means that after maybe particularly 24 days, after 24 days, our compound gives a suppression of 72 percentage in tumor size, whereas particularly standard drug oxaliplatin particularly gives a only 34 to 40 percentage in a particularly H46 and HCT116 cell lines. We have also checked that body weight change of the compounds. There is a no significant body weight change after giving the drug, which is almost similar to the oxyplatin's dose. So there is no body weight change in the mice models. So this suggested that this compound have a very good biological response in vivo study. Here we can see that tumor size is reduced to almost twice than the standard drug. Standard drug gives the 34 percentage, whereas our compound gives a 66 percentage reduced size in a particularly Air Force H46, particularly that non-small cell lung cancer cell lines, as well as HCT116 means that many colon cancer cell lines. Whereas a compound, particularly 72 percentage, is a more 72 percentage tumor size reductions compared with the HCT116 xenograph, which gives only 40 percentage in a standard drugs. So these compounds gives a very good activity in vivo. So this paper we have published particularly in the European Journal of Medicinal Chemistry in 2020. So how drug molecules design, how the particular activity can be achieved through the drug designing approach. So we have seen here the several drug designs for the benzology approach, several drug design for the particularly functional root modifications, several drug design we have particularly through the lipophilicity enhancement. So these all types of work we have published in a very good reputed journals, in the Journal of Medicinal Chemistry as well as European Journal of Medicinal Chemistry. So any questions? Hello. So thank you all. Any questions regarding this? Okay. Hello. Any questions? So if you don't have any question, then we can complete this.